Hesi A2 Biology 2025. Question 1. Which type of bond is formed between amino acids during protein synthesis? Hydrogen bond, peptide bond, ionic bond, disulfide bridge. Peptide bonds form covalent links between amino acids. Hydrogen bonds stabilize secondary structure, ionic bonds occur in side chains, disulfide bridges are between cysteine residues. Question 2. The movement of water across a semi-permeable membrane from low solute concentration to high solute concentration is known as diffusion, facilitated diffusion, osmosis, endocytosis. Osmosis is water movement across a membrane down its concentration gradient, low to high solute. Diffusion moves solutes, facilitated uses carriers, endocytosis engulfs particles. Question 3. What phase of the cell cycle involves the replication of DNA? G1 phase, S phase, G2 phase, M phase. DNA replication occurs during S synthesis phase. G1 is growth, G2 is preparation for mitosis, M phase is division, mitosis/cytokinesis. Question 4. Which cellular organelle is surrounded by a double membrane and contains its own DNA? Ribosome, lysosome, mitochondrion, endoplasmic reticulum. Mitochondria and chloroplasts have double membranes and their own DNA. Ribosomes are non-membranous, lysosomes single membrane, single membrane. Question 5. A mutation that changes a single nucleotide but still codes for the same amino acid is called a what? Missense mutation, nonsense mutation, silent mutation, frameshift mutation. Silent mutations change DNA but not the amino acid due to genetic code degeneracy. Missense changes amino acid, nonsense creates stop codon, frameshift shifts reading frame. Question 6. Which vessel carries oxygenated blood away from the heart to the body tissues? Vein, artery, capillary, venule. Arteries carry oxygenated blood away from heart, except pulmonary artery. Veins return deoxygenated blood, capillaries exchange gases, venules collect from capillaries. Question 7. The final electron acceptor in the electron transport chain during aerobic respiration is which molecule? Water, NAD+, oxygen, carbon dioxide. Oxygen is the final electron acceptor, forming water. Water is a product, NAD+ carries electrons, CO2 is released in Krebs cycle. Question 8. Which process describes the engulfment of large particles by a cell forming a phagosome? Pinocytosis, phagocytosis, exocytosis, receptor-mediated endocytosis. Phagocytosis engulfs large particles, cell eating. Pinocytosis engulfs fluids, cell drinking. Exocytosis secretes, receptor-mediated uses receptors for specific molecules. Question 9. In a DNA molecule, adenine always pairs with which base via 2 hydrogen bonds? Guanine, cytosine, thymine, uracil. Adenine pairs with thymine via 2 hydrogen bonds in DNA. Guanine pairs with cytosine, 3 bonds. Uracil pairs with adenine only in RNA. Question 10. Which organelle is most prominent in cells that produce large amounts of secretory proteins such as pancreatic acinar cells? Rough endoplasmic reticulum. Smooth endoplasmic reticulum. Peroxisome. Nucleolus. Rough ER with ribosomes synthesizes secretory proteins. Smooth ER makes lipids, peroxisomes detoxify, nucleolus makes ribosomes. Question 11. The idea that all living things are composed of cells and that cells arise only from pre-existing cells is called what? Germ theory. Cell theory. Evolution theory. Endosymbiotic theory. Cell theory states cells are basic unit of life and arise from pre-existing cells. Germ theory causes disease, evolution explains change, endosymbiotic explains organelle origins. Question 12. If a red blood cell is placed in a 0.9% NaCl solution, isotonic, what will happen to its volume? Increase dramatically. Decrease dramatically. Remain relatively constant. First swell then shrink. 0.9% NaCl is isotonic to blood, so no net water movement occurs and volume remains stable. Hypotonic causes swelling, hypertonic causes shrinking. Question 13. Which stage of cellular respiration produces the majority of ATP molecules? Glycolysis. Pyruvate oxidation. Krebs cycle. Electron transport chain. Electron transport chain produces 34 ADP via oxidative phosphorylation. Glycolysis nets 2 ADP, Krebs cycle 2 ADP, pyruvate oxidation none directly. Question 14. Alternative splicing of pre-mRNA allows a single gene to code for how many different proteins? Only one. Exactly 2. Multiple different proteins. Non-splicing removes introns only. Alternative splicing joins exons in different combinations, producing multiple protein variants from one gene, increasing proteomic diversity. Question 15. The random distribution of homologous chromosomes during meiosis I is termed what? Crossing over. Independent assortment. Synapsis. Disjunction. Independent assortment shuffles homologous chromosomes randomly to gametes. Crossing over exchanges DNA, synapsis pairs homologs, disjunction separates them. Question 16. Which type of RNA brings specific amino acids to the ribosome during translation? mRNA. tRNA. rRNA. snRNA. tRNA has anticodon and carries specific amino acids to ribosomes. mRNA carries code, rRNA is structural, snRNA processes pre-mRNA. Question 17. A person with blood type AB has which antigens on their red blood cells? Only A antigens. Only B antigens. Neither A nor B antigens. Both A and B antigens. Type AB has both A and B antigens. Type A has only A, type B only B, type O has neither. Question 18. The breakdown of hydrogen peroxide into water and oxygen is catalyzed by which enzyme found in peroxisomes? Catalase. Amylase. Lipase. Protease. Catalase decomposes H2O2 into water and oxygen, preventing oxidative damage. Amylase breaks starch, lipase breaks fats, protease breaks proteins. Question 19. During which phase of mitosis do sister chromatids separate and move to opposite poles? Prophase. Metaphase. Anaphase. Telophase. Anaphase separates sister chromatids pulled by spindle fibers. Prophase condenses, metaphase aligns, telophase reforms nuclei. Question 20. Which organelle modifies, sorts and packages proteins into vesicles for secretion? Rough ER, smooth ER, Golgi apparatus, nucleus. Golgi apparatus modifies, sorts, and packages proteins. Rough ER synthesizes, smooth ER makes lipids, nucleus stores DNA.